So those were genes that are linked to a sex chromosome. So, either an X or a Y. And so that's why they were called sex-linked. Now we're going to talk about something a little bit different. So if you think about how large chromosomes are, except for the Y, each chromosome has hundreds or thousands of genes. And so if we think about genes that are related, well, not related, but genes that are located on the same chromosome, if they're on the same chromosome and are close to each other, they tend to be passed on together, actually. So genes that are located on the same chromosome tend to be inherited together and are called linked genes. So if these are two different genes, but they're on the same chromosome, then they're called linked genes. So they're not sex-linked, they're just genes that are linked to each other, like physically, basically, because they're on the same chromosome. So yeah, genes that are located on the same chromosome are called linked genes. And the closer that the genes are on the same chromosome, so if they're more close together like that, they're more likely to get passed on together versus if they're farther apart on the same chromosome. So they're still linked, but there's different degrees of linkage, basically. And we'll talk about that in a few slides. So Morgan, again, was a super curious guy, and so he did experiments with fruit flies to see how the linkage between different genes affects inheritance of two different characters. So Morgan crossed flies that were different in two different traits, kind of like Mendel did, and so we'll make some comparisons later. But so he crossed flies that were different with two different traits, so body color and wing size. And so when he did that, he saw some interesting differences in the patterns. So let me show a picture really quick. So body color and wing size. So the wild type body color is like a gray color. And then the wing size, this is like the normal wing size. And then, so this is the wild type. And then, so he found flies that were different in both of those things. So he found flies that had a different color, so a black body, and small wings that he called vestigial wings. They were really small, and so they didn't really serve their purpose. They were vestigial. So he crossed flies that were different in both of those things. The body color and then the wing size. And so that first cross was the P generation again for parents. And so that first cross, he made the F1 generation, and those were both dihybrids. So again, they were hybrids in these two different things. So he crossed those to make the F1 flies, and then he crossed those flies to each other and made, he crossed an F1 dihybrid to one that's recessive for both of those traits. So he did a test cross in this case. So instead of crossing these to another one, he was like, okay, well, I want to see how the linkage affects inheritance. And so what if I cross one of these guys that's dihybrid to one that is homozygous recessive for both of these things? So he did a test cross. So yeah, let's track this. He crossed a wild type fly that was homozygous dominant or homozygous wild type for both of these things to a double mutant. So it was homozygous, kind of like recessive, for both of these traits. So black body and then vestigial wings. So again, remember the lack of a plus is like the mutant allele, and then if there is a plus, it's the wild type alleles. So when he crossed these, they all looked basically like this parent, but they were heterozygous. So there was a wild type and a mutant allele, and then a wild type and mutant allele. So they were a dihybrid, a hybrid for two different traits. So yeah, instead of crossing this one with another one of its kind, he was like, well, let's see what happens if I cross it with a homozygous recessive fly. And so let's see what happens. Just again, this helps teach us a little bit about inheritance patterns if we see how one might cover up another trait. So when he crossed these guys, these were the different possible gametes from each one. So if this is the female, then the gamete could have these different combinations. So it could have gotten this chromosome with the wild type for body color and the wild type for wings. Or this other egg could have inherited the mutant allele for each one. Or in this case, because again, an independent assortment, right? This one could have inherited the wild type for body color and then the mutant type for wings. And then lastly, this one could have gotten the last possible combination, which is the mutant allele for the body color and mutant allele or wild type allele for wings. So these are the 4 possible eggs because of independent assortment. Or so we think, right? And then these are the possible gametes or sperm cells from this guy. And basically it can only make this, so that we, again, just shortened the Punnett square by only showing 1 row. If we did 4 rows, it would all be the same thing. So these are the possible combinations from here. Yeah, mutant and mutant. And so in these combinations, in the offspring from this fertilization of this and this, this was heterozygous for both and showed wild type. This one had black body and vestigial wings. This one had the gray body and vestigial wings. And then this last one had the black body and then normal wings. So he found a combination, right? But the ratios were different than what he thought was going to happen. So what could have happened are these 2 different scenarios. So if those genes for body color and wing size were on different chromosomes, then he would see 1, 1, 1, 1 as the ratio. He would see like an even split of these, each of these. If the 2 different genes for these 2 different things, so again, body color or wing size, if they're on the same chromosome, the ratio is going to be different. So it would end up 1 to 1 to 0 to 0. And that's actually what he saw. So he crossed a bunch of these flies. He counted a bunch of them. And what he added them up to was 965 of these guys. Gray, normal wings, 944 of these guys. Black body color, the little vestigial wings. And then he only found about 206 of these guys. Well, not about, like literally 206 of the gray body color and vestigial wings. And then the black body color normal wings, he saw 185 of them. So again, he did this with so many, he was like, this has to be something, right? This is a pattern. I did this cross hundreds of times probably, and this is what I counted. That's not a coincidence. So this ratio, even if it's not perfect, is really close to this ratio, telling me that this means the genes are on the same chromosome, actually. So those resulting flies from the test cross of the F1 hybrids to the homozygous recessive showed totally different patterns than what he expected. The resulting flies had a much higher than expected proportion of the combination of traits that were seen in the P generation. So he concluded that these genes actually don't assort independently. They are linked together because it does happen to be the case that this is going to be passed on with this. This color is going to more likely be passed on with the wing size that's normal. And then these are more likely to be passed on together also, the black body color and vestigial wings. So these traits do seem to get carried along together. So yeah, he concluded that these genes don't assort independently and that they are on the same chromosome. And so again, now that we know, with this knowledge about the chromosome theory of inheritance, we can show, okay, this makes sense. Yeah, they are on the same chromosome. So here is the dihybrid female. So she had one of each. Remember, she was heterozygous for both. And so now that we could picture them like this on the same chromosome, it makes sense. So the black body color. Oh, sorry, the white. Oh my God. Sorry. Okay, so the gray body color is wild type. The normal wings is wild type. So on this chromosome is where these two genes are, the wild type. And then on this chromosome was the mutant alleles. And so when we cross one of these, or when he crossed one of these to a homozygous recessive one that had the wild type on both chromosomes, we can show, okay, this makes sense that they got passed on in that ratio because they're literally like on the same chromosome. So most of the offspring had this or this because they were linked. Like this chromosome did get passed on and had both of these alleles. And then this chromosome did get passed on and had both of these alleles. Or, you know, the other blue one with the light pink one. So these are the two blues that would be the same. And then if these got passed on like this, that is how you ended up with more offspring that had those combinations of traits. This and this, and then, or this and this. Now, what about these guys? How did that happen? We will get to this in a second. So, yeah, now that we can like picture them on the same chromosome, it makes sense. So, the ones that looked like either of those original parents in the P generation are, we call them, like they had parental phenotypes because they looked like the parents in that parental generation, either gray body and long wings, or the black body and vestigial wings. So those were the parental phenotypes. And so, yeah, you saw some of those. Most of them were those. But then there were some other weirdos, basically, right? So the other ones are called non-parental phenotypes. Like they didn't look like either of those parents in the P generation. What is with that? What's with this random combination here? Why wasn't it a perfect 1, 1, 0, 0? And also, now we know it's because of genetic recombination. So remember in meiosis, there's that weird thing called crossing over or recombination. That's how some stuff gets unlinked to each other. So that's how those non-parental phenotypes were made in the test cross. This shows that the two traits could be separated sometimes. So even though most often they are tied together and inherited together, the fact that there were some of these other ones is like, whoa, okay, something else is going on. So this involves genetic recombination, the production of offspring with combinations of traits differing from either parent in the P generation. So, yeah, those findings of Mendel and of Morgan also demonstrated an important aspect of chromosomal basis of recombination. So yeah, offspring that have a phenotype that match either one of the original parental phenotypes are called parental types. And then the offspring that had new combinations that didn't look like those original parental phenotypes, those are called recombinant types, or just recombinants for slightly shorter. So, yeah, when you look at the phenotypes, you basically play like a little matching game. Does it match either of the parents? Then yeah, it's like a parental type, what it looks like. But if it's a new combination that did not look like any of the parents in the P generation, then those are called recombinants. And so I will again show a picture of like crossing over and recombination, but that's what accounts for this. And so a 50% frequency of recombination is observed for any two genes that are on different chromosomes. So even if they're on different chromosomes, they can still get combined to be together. Super interesting. So if we think about Mendel's test crosses, again, first of all, he got super lucky in studying what he did because they were on different chromosomes. I wanted to cut and paste some stuff, but so say, remember he crossed gametes from a yellow round dihybrid parent. Let me just draw it actually. So he had a plant that had perfectly yellow and round seeds. And so they were dihybrid. And so the gametes possible from this dihybrid could have been, big Y, big R, like that. It could have been little y, little r, like that. It could have been the big Y, little r, in this combination, making this gamete, or it could have been the little y, big R, like that. And he crossed those. If he did a test cross, he crossed those with a wrinkled it's not that wrinkled, but whatever. Okay, and it was recessive. So if he did this test cross, then, the only combinations really are little y and then little r. So that's why there's just one. So if he did this test cross and he saw these different combinations of offspring, he saw some that were dihybrid. They were heterozygous for both traits, but showed yellow and round. And this one was green and wrinkled. And so both of these do look like those parents, right? So I was going to copy this and then paste it right here, but you get what I mean with my drawing. So if you look at this one, that looks like one of the parents, yeah. You get this one, this looks like one of the parents, yeah. So these are called parental type offspring because they look like the parents. But these guys have a new combination that wasn't seen in the parents. Neither of the parents were yellow and wrinkled or green and smooth. So that's what I mean, like you call these recombinant because it's a new combination that wasn't seen in the parents. If they don't look like the parents, then it's a recombinant offspring. So, Morgan observed that although some genes are linked, there are non-parental combinations that are still produced somehow. And so he proposed that some process must occasionally break that physical connection between genes that are on the same chromosome. And now we know that mechanism was the crossing over of homologous chromosomes. So remember, in meiosis 1, when the homologous chromosomes are next to each other, actually like this, there is physical exchange of those non-homologous or non-sister chromatids, right? And so that's how you get some that are like part dark blue, part light blue, or part dark red and part light red, because it came from the other part. So that is crossing over, and that's how some of them that even though they were on the same chromosome got unlinked from each other. So if we look at this female fly that has the wild type gray body, normal, and normal wings, so if she was crossed with a double mutant, and these are the F1s, when this female in the F2 or F1 generation goes through meiosis, she might have gametes that have a different combination of those alleles. So this one, if this is her like germ cell that's going to go through meiosis, it replicates the chromosomes. So now there's going to be 2 dark blues. Replicate this chromosome. Now there's 2 dark red and then 2 light red. But then in meiosis 1, when they're physically close together, the homologous chromosomes, there can be that event, that like crossing over, and they literally like break off a piece of this chromosome and put it on this chromosome, and then vice versa. And so this chromatid has a new combination, and this chromatid has a new combination. And then when those get separated, and then the sister chromatids get separated, these are the different possible eggs at the very end of meiosis. So you'll have some that have like a normal chromosome or normal combination of alleles. This one is the normal wild type or mutant combination of alleles, but then this is a new combination. So these are recombinant chromosomes. They recombined those alleles. And so that is how you get the recombinant phenotypes, recombinant offspring that have a new combination that wasn't tied together. And so again, he combined all of his data and looked at the big picture pattern, and he found that there was a recombination frequency of like, okay, if there's this many offspring total, and this many of them are new recombinants, then that means that there's like about a 17% recombination frequency out of all of the total offspring. This is how often like this recombination thing happens between those 2 different genes. So, recombinant chromosomes bring different alleles together in new combinations in gametes. So that's how, So recombinant chromosomes bring those 2 different alleles for the 2 different genes together in new combinations in gametes. And the fact that this could happen in other flies if there's random fertilization and you're not doing like specific crosses, then that means there's like infinite number of combinations produced in offspring. So random fertilization, again, is super huge in giving new variation, because this is happening, recombination is happening in the egg cells. And then it also is going to be happening in sperm cells, just not in the ones that we crossed here purposefully because they are homozygous recessive. But if any fly was allowed to mate with any fly, like all these different combinations, is so interesting. And again, that is why, like, you and your siblings don't look exactly alike, and you and your siblings probably don't look exactly like your parents, because all of our chromosomes are doing this stuff, this recombination. And so if each of the egg cells has like a new combination possibly, and each of the sperm cells could possibly have a new combination, and random fertilization, like that's why, yeah, you don't look exactly like your parents. And neither do your siblings, and you don't look exactly like them because each of you get like a unique blend of alleles. And yeah, so it's super interesting. And so this abundance of genetic variation is the raw material that natural selection works on. Like, the more variation that there is in a population, the better, because changes in an environment will end up having more different things that could benefit, if that makes sense. So yeah, if you think back on like natural selection, and like populations of organisms being so different, it's a lot better for populations to have this variation,